At this point, they apply a carefully timed pulse of light to split the sphere's quantum state, placing it into a superposition of being in two slightly different positions simultaneously. They have successfully created the space-time blister that Penrose's theory describes, but with a glass bead instead of a tubulin protein. Now, the critical observation begins. The experiment is a waiting game. The physicists monitor the sphere's position, looking for the telltale signature of a spontaneous collapse. According to Penrose's equation, given the mass of the nanosphere and the separation distance they created, they can calculate the predicted lifetime tau. For a typical experiment, this might be on the order of milliseconds to seconds. If, after many trials, they observe that the superpositions consistently decay at or around this predicted time, it would be groundbreaking evidence for objective reduction. It would show that gravity does indeed play a role in resolving quantum uncertainty. These experiments are at the absolute cutting edge of modern physics. They are incredibly difficult to perform, as they require pushing technology to its limits to fight against every possible source of environmental decoherence. However, they offer a clean, unambiguous path to testing the new physics proposed by Penrose. Unlike the messy biological experiment, there are far fewer confounding variables. The mass of the nanosphere is known precisely, the vacuum is controlled, and the temperature is managed. The system is designed from the ground up to be a perfect testbed for fundamental physics. The results of these levitated particle experiments will have profound implications for our original question. If they fail, if the superpositions last much longer than Penrose predicts and only decay due to known sources of decoherence, then the physical foundation of OR would crumble. But if these experiments succeed, if they confirm the gravity-induced collapse model in a non-biological system, the situation changes dramatically. It would prove that the physical mechanism required by OR is real. The debate would then shift back to biology, but with a crucial new piece of evidence, the spooky physical process required for quantum consciousness would have been shown to be a genuine feature of our universe. The stage is set for the definitive biological experiment where quantum gravity meets neuroscience in the lab. Every step is pre-registered and transparent, locking in the protocol before a single neuron is touched. We use cultured neuronal networks or rodent brain slices, split into control and test groups, with every variable tightly controlled. Double-blind procedures and coded samples ensure unbiased results. Multiple control conditions, including unrelated isotopic substitutions and microtubule targeting drugs, guard against confounds. Only with this fortress of rigor can we trust what the experiment reveals. The experiment's heart is isotopic labeling, feeding neurons amino acids with heavy carbon-13 for the test group and standard carbon-12 for controls. Ideally, the heavy isotope targets only the microtubule regions implicated by OR, isolating the mass change. Mass spectrometry confirms the precise incorporation of isotopes, ensuring no unintended cellular changes. Neuronal health and function are checked. If basic properties differ, the experiment fails. Only when both groups are identical, except for microtubule mass, do we proceed. Now, we search for the signature of consciousness. With verified cultures, we stimulate neural networks and record their electrical activity. Microelectrode arrays capture gamma band oscillations, OR's predicted signature. We analyze hundreds of trials, measuring the exact gamma frequency for each. The goal: detect a subtle, systematic shift in frequency between groups. Secondary measures, amplitude and network connectivity, help interpret results.